Hello, my lovely anatomists and physiologists. Michele Glass here. Our conversation for this video is hemopoiesis or hematopoiesis. You'll see it both ways. This prefix heme is talking about blood, and then this term poiesis is talking about formation of. So this term is talking about the making of blood cells, and we're talking then about all of the different formed elements. So we do start here with a stem cell called hemocytoblast. And stem cells, remember, are cells that maintain their ability to do mitosis. Mitosis is talking about cell division. And we do see that these hemocytoblasts are going to be located in the red bone marrow, which can be in the medullary cavity of long bones, in the developing human, but then we'll see as adults, this is more in the spongy bone of the epiphyses of those long bones. And when we talk about a stem cell maintaining the ability to do mitosis, remember when we do mitosis, we divide and make two cell types. And so as part of this mitotic division, we see that the hemocytoblast makes another hemocytoblast. So it's maintaining this ability, it's remaking itself, right? So it's maintaining these stem cells. When the hemocytoblast is doing mitosis and dividing, we'll see that, of course, it remakes a cell so that it can continue this process. And then it will either make what's referred to as a lymphoid stem cell, or it will make what's called a myeloid stem cell. And we'll see that another name for that red bone marrow is myeloid tissue. So it's being named after this type of cell. Now, the lymphoid stem cell is going to specifically give rise to lymphocytes. And lymphocytes are one type of white blood cell. So we talked about there are 5 types of white blood cells, and the lymphocyte is just one of those 5. Now, the lymphocyte we'll see is really important in our immune response. So we will be talking about them more in the future in the lymphatic system. But we will see that the lymphocyte, there are 3 types of lymphocytes. We're going to talk about in the future the NK cells. We'll talk about the T cells. You've probably heard about these before. And we'll talk about B cells. And so when we do our leukocyte, when we do our white blood cell video, we'll go into a little bit of detail here. And then when we study immunity in our lymphatic system, we'll go into a lot of detail there. Looking back at our myeloid stem cell, if we've produced this, then the myeloid stem cell is going to give rise to 3 different cell lineages. So the myeloid stem cell will give rise to what's called a megakaryocyte, mega big, karyonucleus site cell. So it's a big nucleated cell. And what we'll see is the megakaryocyte will break down into fragments, and we call these fragments platelets. Now we will see that there's a hormone that's involved in this process, and we'll talk about this hormone as thrombopoietin. So thrombopoietin is going to be produced by the liver and the kidney. And it's going to trigger the production of platelets. We also see that the myeloid stem cell is going to give rise to our white blood cells. Now remember, we have 5 types of white blood cells. 4 of them are related back to this myeloid stem cell. So when we talk about these white blood cells here, we can list out, you can have the neutrophil, You can have the basophil, you can have the eosinophil, and you can have the monocyte. And so the white blood cells coming from the myeloid stem cell, you can have any of these four, whereas notice the lymphocyte has its own stem cell lineage. Now, just like we had some hormone and chemical signals involved in this development, we're going to see cytokines here, and we're just going to talk more about these in the future. So just kind of like mark this as a placeholder, and we'll come back to that. And then the last type of cell that we'll get, and I'm going to show right here like a few arrows because there's lots of steps, and I'm going to do those on a different screen, is the red blood cell. And this process of making a red blood cell has a name. It's called erythropoiesis. And it is going to be coordinated by a hormone called erythropoietin. Oh, let me change my pen color. I always want to forget my Y when I spell out erythro anything. We can abbreviate this hormone erythropoietin as EPO. And what we'll see is that this is going to be produced by the kidney. It's going to be produced when you have a low blood pressure or low oxygen levels. And it's going to stimulate an increase in the production of red blood cells. This process of erythropoiesis, we need to know in a little bit more detail. So let's go ahead and do that next. Okay, so remember we started in the red bone marrow with the hemocytoblast, and then that divided to give rise to the myeloid stem cell. And the myeloid stem cell is giving rise to all of the formed elements in the blood except lymphocytes. And so here we're focusing in on this process of erythropoiesis, which is going to be the formation of red blood cell. Red blood cells, right? And remember, we already mentioned that this is going to be under the control of erythropoietin, which we can abbreviate as EPO. Now, what we see happening is in the red bone marrow, the myeloid stem cell under the control of the erythropoiesis is going to produce a cell that's called the pro-erythroblast. So a blast cell is a producing cell. It's making something, and pro here is becoming what we're going to see as an erythroblast. So this is like day one. And then we see it's developing into the erythroblast. And what we should consider is that each one of these names is more telling us something about where we are in the development. It's telling us something about how far along the maturation is of the cell. So it's kind of this idea of, you know, in humans, we call you, you know, an infant from like 0 to 1. And then once you start kind of moving around in space, walking around, you're getting into the, you know, 1 to 2 or 3 years old, we start calling you a toddler, right? And then a preschooler, and then you're like a school-aged kid, and then you're a tween, right? And then you're a teen, and then a young adult, and so on. And so what that's really telling us is something about where you are in your development, right? And so that's what we're doing with these different names. We're talking about the same cell essentially, but it's changing in terms of where it is in its development and where it is in its preparation to be this future red blood cell. And then we have a cell that we call the normoblast. So by around day 4, our development is progressed enough that we call the cell a normoblast. And what's really happening in this whole period of time is we're making hemoglobin. And hemoglobin, we'll see, is the molecule that actually transports the oxygen gas and the carbon dioxide gas. And this time we're making like 80% of the hemoglobin that this future red blood cell is going to need. So, you know, when we're start out day 1, it's a proerythroblast. And as it starts developing and making more and more hemoglobin, it's an erythroblast. And then it continues in this maturation process, and then it's called a normoblast. And then we get to what's called a reticulocyte. And this is showing up at about day 5 to 7. And the significance of this is that it's going to eject the nucleus and most of the organelles. It's going to eject the nucleus and most of the organelles. And so we talked about, why are our formed elements called formed elements and not called cells? This is one of those reasons. When we look at a mature red blood cell, it has no nucleus, it has no endoplasmic reticulum, it has no mitochondria, it has no Golgi apparatus. And so what that means is it becomes very limited in its ability to make and repair proteins. And so the reason the proerythroblast, erythroblast, and normoblast is really heavily making this hemoglobin is because when it gets to this reticulocyte stage, it's real limited. It's just like finishing up the last little bit. It kicks out the instructions. So mRNA from transcription is present in the cell for 24 to 48 hours. And so while you still have mRNA, you can continue to make hemoglobin. And once that mRNA degrades, the nucleus is gone. There's no more instructions. And so you're finished in your making of hemoglobin. And you're also now like super limited in your ability to repair. And so then what we see happening is the reticulocyte is what's going to actually enter circulation. And it's going to be in circulation for about 24 hours. And that's when we then call it, let me go ahead and say erythrocyte, or what we can abbreviate as the red blood cell, RBC. I guess I'm just talking like one, so let me make that singular. And so then we have our mature erythrocyte in our circulation. And so that then is our topic for our next video. So stay tuned. And as always, take care of yourselves and each other.